Hello, and welcome to another episode of the Science on the Edge podcast. In this episode, we're going to be talking about the immune system. So this is a deep dive into human immunity. We're going to talk about both the passive and the active immune system. I thought it was quite a good idea to put together a podcast on this topic with the pandemic booming. I thought it may help you guys to have a bit of background knowledge as to exactly how your immune system fights infections. So I'm going to do a pretty deep dive into this topic today. Before I get started, I'd just like to say that anyone who supports me via Patreon or via PayPal by making a donation on my website, that's much appreciated. Every little donation helps me keep motivated. If you'd like to donate, then simply go to scienceontheedge.com and click on the donate button. Or alternatively, you can go to Patreon and go to Science on the Edge on Patreon and donate that way. Okay, but that's enough of that for now. Let's now do the deep dive that was promised into the immune system. So let's get started. Okay, so the immune system by myself, Dr. Mark Darcy. Okay, so let's start at the beginning. What is the immune system, and what is it evolved to do? Well, it's actually evolved to do quite a few things. It's not just about fighting infection, it's about protecting our body from all sorts of natural toxins which exist in the environment. The world is full of toxins produced by all sorts of organisms and produced naturally, and our body needs to be able to cope with those toxins. So why have a defense system? Well, to deal with potentially harmful bacteria, virus, etc., that are continually trying to infiltrate our systems. And if we do not destroy such infiltrators, then disease or even death may result. You may recognize this particular virus here. This is COVID-19. Particularly nasty little bug. Okay, so our defense systems. We have the non-immunological defense systems. This basically prevents entry of harmful substances. So it comprises of things such as physical barriers. So mucous membranes in our lungs and our intestine, for example, and the skin on the surface of our body. We also have chemical substances that we release, lysozymes, which are these little chemicals that we release on the surface of our skin and in other parts of our body to just digest and neutralize various offending organisms, viruses, bacteria, fungi, etc. Then there's the immunological defense system. This removes harmful substances from our body. There's the internal innate immune system, and then there is the adaptive acquired immune system. I'm going to go into both of those different systems in quite a bit of depth with some repetition just to make sure that the concepts do sink in. So let's get started with that. So first of all, the lymphatic system. What is the lymphatic system? Well, it's a network of vessels throughout our body, vessels and tissues, that's kind of home to our immune system, helps protect us. We have a blood system, a circulatory system, so blood vessels going throughout our body, but we also have lymphatic vessels, a lymphatic system going up throughout our body too. And it's a sort of highway for immune cells. So the immune system has two physical components, the lymphoid tissue and cells responsible for the immune response. The lymphatic system includes lymph, which is a kind of white, kind of creamy substance that you find in your lymphatic system, and lymph nodes. Lymphocytes, which are basically a type of white blood cell. There are T lymphocytes and B lymphocytes. They're part of the adaptive immune system. That's the part of our immune system that can learn from past encounters, can make antibodies, that kind of thing. And there are the lymphatic vessels. There's also lymph nodes. You felt lymph nodes, your tonsils are lymph nodes, your adenoids are lymph nodes, there are lymph nodes under your armpits and your groin and throughout your body. These are they're parts of the lymphatic system. The way to think about it is if your lymphatic vessels are kind of like roadways, then the nodes are kind of like little towns or cities along the roadway. And they're a site where lots of immune cells can get together and be introduced to any viruses or bacteria that have invaded the system, so that a good, strong immune response can be made. Tonsils are basically just large lymph nodes. The spleen is where the lymphatic system comes into very close contact with the circulatory system. So there is blood and there is lymph in very close contact. In case you're wondering what the spleen is for, a couple of things. One thing that it's for is basically getting rid of damaged blood cells. So any blood cells that are old and damaged, they will be looked at by the immune system in the spleen and removed if found to be harmful. The thymus gland. Thymus gland is about here. Babies and children have very large thymus gland. It's where your immune system, it's where your adaptive immune system kind of matures and develops and learns about infection. As you get older, the thymus gland does shrink, though, quite substantially. And a lot of what's in the thymus gland as you get older doesn't really work properly, which is probably part of the reason we do get old. As our thymus gland starts to degenerate, our ability to mount an effective immune response also starts to degenerate. So the lymphatic system helps to maintain fluid balance in tissue. It absorbs fats from the digestive tract. So any fat that you eat is, first of all, emulsified by bile from your liver. And then it's turned into very small droplets. And those droplets are absorbed into the lymphatic system and move around your body through that system. It forms part of the body's defense system against microorganisms and other harmful substances. Two primary lymph tissues are the thymus gland and the bone marrow. T lymphocytes, T stands for thymus. Those particular lymphocytes, immune cells, mature in the thymus. B lymphocytes, so B stands for bone. The B lymphocytes, which make antibodies and release them, they basically are formed in the bone marrow. At both sites, cells involved with the immune system are formed and mature. There's also secondary lymphoid tissue, sites where mature immune cells interact with pathogens, initiating an immune response. All sorts of tissues such as lymph nodes, tonsils, adenoids, etc. And here's an overview of the system. As you can see, all of these vessels throughout the vessels you're seeing here, these green vessels, not actually green, but these green vessels are the lymphatic system that flows around the body with various lymph nodes at different points. The thymus, as you can see in the chest. We also have the adenoids and the tonsils. We have the right lymphatic duct, which is kind of where a lot of the lymph empties out into our bloodstream. We have the spleen there, another important lymphatic organ. And as you can see, when you zoom in on a lymph node, and there on the right, you can see that we have blood vessels coming in very close contact with lymphatic vessels. So there can be a sort of exchange at those points. So lymph tissues, secondary lymph tissue is divided into encapsulated diffuse lymph tissue and unencapsulated diffuse lymph tissue. Encapsulated lymph tissue includes the spleen and lymph nodes. Both have outer walls formed from fibrous collagen you have under your skin, slightly different kind of collagen in the lymph nodes. It's a very fibrous collagen tissue. These are capsules. The spleen contains immune cells known as phagocytes, from the Greek to eat. So phagocytosis is when a cell eats another cell, as our white blood cells do with invaders such as bacteria. So phagocytic cells also trap and remove aging blood cells too, and other things as well in our body. Lymph nodes contain clusters of immune cells. They intercept pathogens entering through the interstitial fluid. That's a fluid that surrounds our organs. Our organs are not bathed in blood. We have blood vessels going through our organs, and liquid leaks through the blood vessels, through the walls of the blood vessels, into the tissue. And it's that liquid that bathes our tissues, and that liquid is known as interstitial fluid. It contains various nutrients, oxygen, etc. So lymph nodes contain clusters of immune cells. They intercept pathogens entering through interstitial fluid, either through breaks in the skin or through mucous membranes. So for example, in our nose, in our bronchial tubes, in our intestines, that kind of thing. The unencapsulated lymph tissue include the tonsils, the skin, the respiratory, urinary, and reproductive tracts. Immune cells positioned there to intercept pathogens before they get into general circulation. Primary cells of the immune system. I'm going to talk about these a little more as we progress, but I'll give you an overview now. Cells of the immune system are found in the highest concentrations wherever they are likely to encounter antigens that penetrate the epithelia, meaning surface layers such as skin. Primary cells of the immune system are divided into 6 categories. First of all, there are the eosinophils, sometimes known as acidophils. They fight parasites and contribute to the allergic reaction. Basophils in the blood and mast cells, these release histamines and other chemicals. The neutrophils, these eat bacteria and release cytokines. So they eat bacteria through phagocytosis and they release cytokines. Cytokines are chemical messengers that can alert the immune system that there's some sort of infection and get everything primed to deal with that infection. The monocytes, they mature into what we call macrophages. So they're made in the bone marrow. Then they move through the blood to different sites around our body, all the different tissues. And when they set at home in a particular tissue, they then become a macrophage. So if you think of neutrophils as kind of like patrolling soldiers that are just moving around the body looking for something to fight, that's a neutrophil. Whereas a macrophage is kind of like a guard. It sort of positions itself in some tissue and just sits there waiting for an attack. I should say that macrophages are also phagocytes. They can eat bacteria as well. And they also release cytokines, such as same as the neutrophils, so they can trigger the immune system to become active too. There's lymphocytes and plasma cells. These mediate the acquired immunity. So lymphocytes such as B lymphocytes, T lymphocytes, and plasma cells, which mature from B lymphocytes, they basically have antibodies. They can either have them on their surface that are specific for specific bacteria and viruses, or they can release the antibodies, depending on the type. Then we have the dendritic cells. They activate the lymphocytes. I'm going to talk about this in more depth. Basically, cells such as macrophages, when they eat a particular bacteria, they can then present the antigens from the surface of that bacteria to dendritic cells. And then the dendritic cells can then activate the active immune system. So as the dendritic cells can basically show these antigens to the active immune system and therefore stimulate it to become active. I mean, that's a simplification of the process, but in a roundabout sort of way, that's what happens. Okay, lines of defense. So 3 lines of defense that our bodies have. First, physical and chemical barriers, as I mentioned earlier. Intact skin and lining of body cavities and tubes, plus various secretions. For example, in the vagina, certain substances are secreted that are acidic, and they will destroy bacteria if it tries to invade, or at least it will try to. Second, internal innate system, including macrophages and neutrophils. This is a generalized response. Macrophages and neutrophils, they just eat stuff. They don't discriminate between different types of infection. They have no memory. All they do is they patrol looking for anything that doesn't seem like it should be there, and they eat it. This is the innate immune system. Even amphibians have an innate immune system. Then there's the acquired immune system, which all mammals have. The adaptive acquired immune system, this is a specific response. This is where we make antibodies that target very specific strains of bacteria or viruses or toxins. The acquired immune system sounds so much better than the innate immune system. So you might think, why don't we just have an acquired immune system? Well, the reason is it's much slower than the innate immune system. Macrophages and neutrophils are quick. They're not very accurate, and they can cause collateral damage when they attack bacteria. They can damage tissues, but at least they're quick. And when there's an infection in your body, especially before we had medicine, quick was key. The adaptive immune system, however, it's slow. It can take a while to become active, but when it is active, you make antibodies that are very specific. You make smart bombs. And plus, the adaptive immune system has a memory, meaning that you get the infection is defeated, but these little memory cells remain waiting for the infection to come back. And if you encounter that same infection maybe years later, your adaptive immune system is ready, and it will suddenly make lots of clones of the cells that made the initial antibody that attacked successfully that particular infection, and it will be dealt with maybe before you even get a symptom. So physical barriers, skin, mucus, secretions, sweat, saliva, tears, contain antibacterial enzymes. We have cilia, little hairs, for example, in the lining of our bronchial tract that basically can beat upwards to move any toxins or any hazardous substances up out of our lungs. We have flowing liquids such as tears and urine, which can pump out anything that we'd rather wasn't in our body. And all these things work together as a physical barrier to protect us, first line of defense. Chemical barriers, so acid, I mentioned already, complement of various chemicals which can sort of, well, in a basic sort of way, they tag infectious agents such as bacteria for destruction. I'll get into that a little bit later. Interferon, chemical that can interfere with certain processes and again protect you from infection. Antibacterial secretion, sebum, which creates a sort of toxic barrier to infections. Lysozyme, which is found in saliva and tears. It's an enzyme that destroys microorganisms. All of these things act as chemical barriers. Specific chemicals, complement. There's about 30 complement proteins, more or less. And these complement proteins can directly kill cells, or they can stimulate the lymphocytes and attract them to the area of infection. Cytokines, huge number. These are just chemical messengers. They have a wide range of activity. For example, one class, the interleukins, cause inflammation, fever. T and B cells start dividing, stimulates bone marrow, stem cells, attract phagocytes, and activate a type of cell known as a natural killer cell. A cytotoxic cell, which can destroy infected cells if one of your cells has got a virus in it, for example. Interferons, they defend against viruses and activate some lymphocytes. And then tumor necrosis factor, TNF, which triggers inflammation and can kill tumor cells. Okay, white blood cells. So we've talked about a few of them: monocytes, eosinophils, basophils, lymphocytes, neutrophils. Now, they all look very different under a microscope, different shapes, and different functions. So neutrophils, they pretty much just eat things and release a few chemical messengers. Basophils release enzymes. Mast cells, they're involved in a few processes, but they're involved in things such as inflammation, for example. Monocytes and macrophages. Monocytes mature into macrophages and defend local tissues from infection. The eosinophils, they target worms, fungi, and larger invaders. So you can imagine there's quite a few of them in our intestinal tract. Throughout most of history, humans were very prone to intestinal worms and other parasites, and the eosinophils help defend us against them. In fact, it seems that we might have even needed some of these parasites There's a lot more bowel conditions that have developed as we've got rid of these parasites. And it's been shown in some cases that being infected with, for example, intestinal worms can lower the symptoms of things like Crohn's disease and other intestinal complaints. It's sort of a little bit symbiotic with some of these creatures. I mean, don't get me wrong, I'd rather not get infected with intestinal worms, but it seems that in the past they did have a function. The eosinophils, maybe, well, when they're not dealing with what they should be dealing with, maybe they cause other problems. Dendritic cells, I mentioned, lymphocytes, B and T, produce antibodies, etc., involved in the adaptive immune system. And the natural killer cells, which can basically sniff out infected cells. So if you've got a cell that's infected, they can sniff them out, they can get rid of them. Now, what you're seeing here on the right is a sort of family tree of the immune cells. Stem cells are what we call mother cells. They can divide to make all sorts of different types of cell. Now, in our bone marrow, we have a special type of stem cell called an hematopoietic stem cell. And these stem cells give rise to all the different types of blood cells. And you can see many types listed in that tree. You get a bone marrow transplant, you get your own bone marrow destroyed in a bone marrow transplant given, you have an entire new immune system. Okay, so now let's talk a little bit about the innate immune system before zooming in a little later. The internal innate immune system is dependent on white blood cells. Phagocytotic cells such as neutrophils and macrophages, they engulf foreign cells, molecules, and debris. And then the natural killer cells, the NK cells, recognize virus-infected cells and cancer cells. I didn't mention that earlier. If a cell becomes cancerous, there's likely to be some giveaway sign. Certain aberrant proteins will find their way to the surface of the cell, for example. And when they are sniffed, these cells, by natural killer cells, quite often they're killed. We all develop cancer many times in our life, but usually it's dealt with at a very, very early stage by these natural killer cells. In fact, one of the reasons why older people are so much more prone to cancer than younger people is because our natural killer cells and the rest of our immune system becomes weaker with age, so less able to recognize damaged and cancerous cells. And the natural killer cells then release various chemicals. Well, it's a little bit more complicated than that, but they can trigger processes such as apoptosis, for example, which causes a cell to kind of commit suicide if it's infected. There are various defense proteins, so they can, in some cases, destroy infections, prevent division of harmful viruses, such as interferon, trigger an inflammatory response. The purpose of the inflammatory response is basically to put a beacon up in part of your body saying there is an infection. And this activates your immune system throughout your body to deal with that infection. So inflammation is quite important, although inflammation can be overdone. Inflammation causes so much collateral damage. It doesn't just kill bacteria and other infections, it can kill you if it gets out of hand. In fact, as you age, inflammation gradually increases and increases, and an old body is usually an inflamed body. So inflammation is good for sort of triggering the immune system, but if it's overactive, which tends to happen as we get older, there can be lots of collateral damage. Okay, so the internal innate immune system performs phagocytosis, neutrophils, macrophages, they engulf the invader. Inflammation, a series of reactions that gives fast, localized response. Usually you're infected locally, you get a cut, there's an infection locally. So you need a localized response, which is why inflammation tends to be local, although it can become systemic, which is very dangerous. Complement proteins, we mentioned those 30 complement proteins, generally that is, and they can either destroy bacteria directly or infectious agents, or they can attract the immune system. Fever. Our body's temperature is controlled by the hypothalamus, which is part of our brain. The hypothalamus does lots of things, but one of the things it does is it acts as our body's thermostat. If we get an infection, then the best thing to do while our immune system becomes active, especially our adaptive immune system, which can take some time, is to just try and burn out the infection. Bacteria, usually bacterial cells, are not as tough as our cells. So if we up the thermostat, we can slow down the development of these bacteria, we can slow down the reproduction, we can slow down the progression, maybe even kill the bacteria, while our immune system becomes active. That's what a fever is for. We raise our body temperature to try and burn out the bacteria. And fever, in Paleolithic times, was almost, the best course of action, because we didn't have antibiotics and those sorts of things. So it was a very effective response. Unfortunately, if your fever is too high, it can kill you. The idea of fever is to kill the bacteria before the fever or the bacteria kill you. So phagocytosis means eating. So a cell such as a neutrophil or a macrophage will adhere to a particular pathogenic cell using various receptors on the surface of it. The pathogen is often risk marked by molecules known as opsonins to aid recognition. These little molecules get sort of deposited on the surface of infectious agents such as bacteria and mark them for deletion, let's say. The pathogen is then engulfed by the phagocyte. It then fuses this. So imagine the phagocyte will grab hold of a bacteria, which is much smaller than a phagocyte. It will internalize that bacteria, and then it will fuse with the stomach of the white blood cell, the stomach of the macrophage or neutrophil, which is known as a lysosome. And the lysosome is full of lots of nasty chemicals like hydrogen peroxide, bleach, which will then digest the bacteria. So here you can see the process. The pathogen is taken in to a vesicle, fuses with the lysosome, now becomes basically a phagolysosome. It's degraded, and all the rubbish is spat out the other end. That's the innate immune system. Inflammation is quite important here as well. So when it comes to inflammation, cardinal signs of inflammation, redness caused by an influx of blood. So blood vessels dilate and become leaky. So more blood leaks into the site of infection, carrying white blood cells, for example, to fight the infection, and also putting a bit of pressure in there so that it's harder for any invading organism to move into the body. Swelling, influx of fluid. Again, that again is to try and create a barrier to the infectious agent. Heat caused by influx of blood. Pain because of damage to nerves, sensitization by chemicals, pressure, and loss of function. It's a fast local response to an infection. For example, if you get a cut. It's general, so it's non-specific. It just causes swelling in that area and attracts immune cells to the area. It occurs in response to tissue damage or invasion by bacteria. There are three basic stages: vasodilation, the widening of the blood vessels in the area, and increased permeability of those blood vessels. They become leaky so that white blood cells can leave the blood and get into that tissue. Emigration, basically, of the various cells, and then tissue repair. Requires cytokines for activation. There are many inflammatory cytokines, and chemoattractants are released in the local area to attract immune cells. As I mentioned, there are macrophages all through your body, and if they detect some infection, they will release all sorts of chemical attractants to attract other cells to the area to help fight the infection. Complement, cascade reaction involving proteins that results in death of microbes by phagocytosis. Cytolysis, which means the popping of a cell, so you can cause bacteria to pop, for example, and inflammation. Classical pathway involves antibodies and complement proteins. Alternative pathway, activation of complement proteins directly. Fever, abnormally high body temperature. occurs an infection and inflammation due to bacterial toxins. Release of cytokines such as interleukin-1 from macrophages helps trigger this process. That gets to the hypothalamus, and basically the hypothalamus will then dial up the thermostat. Result intensifies the effect of interferons, inhibits growth of microbes, and speeds up body reactions that aid repair of the wound. You need that wound repaired so that no other infectious agent can get in there. So now the adaptive immune system. Adaptive immunity, an overview. So it's a specific response to some antigen. Antigens are basically just little markers on the surface of cells. Every bacteria has its own markers, its own antigens, its own unique ones. Each T or B cell, T or B lymphocyte, makes receptors for only one antigen. We have millions and millions of these B and T cells, and they all make different receptors, basically different antibodies. Every one of them has a different shaped antibody, which will bind to a different type of antigen. There are many, many millions of types of bacteria, so we need many millions of types of antibody, of receptor on the surface of the B and T cells to become specific to these antigens. So we have many millions of antibodies for many millions of antigens. Thing is, though, we don't have at any one time many B or T cells that are specific for any individual bacteria, which is why when you get infected, it's going to take a while for the right B or T cell to encounter those antigens and become activated. This is where things like dendritic cells get involved to trigger them. It takes time. When this happens, by the way, when a particular T or B cell with the right antibody finally does encounter the correct antigen, it grows, it divides, makes many clones of itself, and makes many, many antibodies to flood your system. So there's a diversity. Collectively, T and B cells may have receptors for millions of different threats, millions of types of bacteria and other infection. One of the reasons you have a fever is to slow down the growth of a bacteria while we find the right T or B cell. Memory. Some B and T cells are made, basically they become memory cells. If you've been infected once, then some of the clones of the B or T cells with the right receptor, the right antibody, will become memory cells, and they'll sit in your tissue waiting for the infection to come back, so that if you do get infected again, the response will be really quick, maybe before you even get symptoms. So B and T lymphocytes form in the bone marrow. T cells are then processed in the thymus gland in the chest. They then become helper T cells, cytotoxic T cells. B cells produce antibody to antigen, lots of antibody to specific invading substances. This is known as antibody-mediated immunity. There are cytotoxic T cells which attack invaders directly. Helper T cells are involved in both of these responses. So they helper T cells help different cells in the immune system to coordinate their activity. This is one of the reasons why HIV is such a nasty infection, because HIV basically, it infects T cells, so things like helper T cells. And when it infects them and becomes AIDS, then it destroys those helper T cells. And without helper T cells, you can't coordinate your immune response. And therefore, you're vulnerable to any passing infection. So the B and T cells are stored in the lymphatic system until needed. So adaptive immunity first needs to recognize the invader. Second, clone the responders, make many copies of the various adaptive cell, T or B cell, that basically has the right antibody. Third, respond to the invaders. And fourth, remember the invader by creating memory cells. So recognizing the invader, B and T cells have thousands of receptors on the surface for one specific antigen, work in different ways, these receptors. So antibodies. antibodies are basically the receptors we're talking about. So there are different categories of antibodies, IgA, D, E, G, M. I'm not going to go into too much detail, but they have slightly different roles. So IgA, directly associated with mucosal surfaces, secreted in saliva, tears, breast milk to give the baby an immune system. A baby doesn't have an immune system when it's born. It's not developed yet. So the IgA in the mother's milk is very helpful. IgD can help with the helper T cells. IgE is important in allergic reactions, triggers inflammation after attacks by parasitic worms and other pathogens. That was its original role. Now it's got nothing else to do, so it spends its time causing allergic reactions to phantom pathogens such as pollen. IgG, basically IgG does a few things. It activates complement, for example, and it can cross the placenta to help protect the baby in the womb. IgM is good for dealing with basically viral particles and that kind of thing. So these different antibodies released by the adaptive immune system have different functions, released by different types of cell in the adaptive immune system. This is an antibody, what it looks like. It's kind of like a Y shape, although this is very generalized. Basically, if you look at the antigen binding site there, if you're actually watching this on YouTube rather than listening to it via a podcast catcher, the antigen binding sites, they vary on an antibody. An antibody is sort of like a Y-shaped structure, and it binds to very different things. The structure varies at the end. So antigen binds to antibodies. Specific antibodies recognize specific antigens, and they can bind and help process the immune system. So cell-mediated responses, T cells. So they react against pathogens that hide in cells. Antigen-presenting cells, APCs, present antigen to a T cell. So basically, an antigen-presenting cell is a cell that's come into contact with the antigen. So for example, a macrophage might eat some bacteria, and then it might present some of that antigen to another cell, such as a T cell, which can activate that T cell, which then divides basically divides and can then help process and activate the immune system. All have T cell receptors that recognize the same antigen, so they'll proliferate. So you'll get one T cell that recognizes a particular antigen. It's shown an antigen, recognizes it, will proliferate. So T helper cell response, activated by antigen presenting cell binding, divide to become effector, which basically means they're no effector cells. They are actually doing a job rather than just mediating the immune system. And some become memory cells which will basically sit around until the infection occurs again. Effector T helper cells stimulate B cells and cytotoxic T cells. So once you've got your effector helper T cells, they will stimulate cytotoxic T cells to kill cells and B cells to make and release antibody. So here's an image if you're watching this on YouTube rather than listening on a podcast catcher, but I'll describe it anyway. Helper T cell activation and action. So one, there's antigen recognition. So there's some sort of antigen presenting cell which has got some antigen in it. It will show that to helper T cells. The helper T cells, when become activated, will divide and into two populations: a population of effector cells, which can then do all sorts of things, release all sorts of chemicals, activate all sorts of other cells of the immune system, and then memory cells, which will sit around waiting for an infection to return, and then they'll be ready to activate the immune system once again very quickly. So cytotoxic T cells, activated by helper cell, they bind to infected cell that displays a particular antigen, cause the release of proteins, they enter the infected cell and trigger death of the cell. So let's delve into specific immunity a little bit more. So specific immune system, this system, also known as active or acquired immunity, is slower than the non-specific responses, can take up to 14 days to respond effectively to pathogen invasion. Immune memory cells means that it reacts quickly to a second invasion of the pathogen. Antibodies, these Y-shaped glycoproteins made of protein and sugar, are known as immunoglobulins. They bind to a specific antigen on a pathogen or toxin that triggers the immune response. Millions of different antibodies, a specific antibody for each potential antigen, which is why we can deal with so many different infections. Antibodies are made up of two identical long polypeptide chains, meaning long proteins, two heavy chains, and also two smaller identical chains known as the light chains. Chains are held together by certain bridges known as disulfide bridges, which we won't go into right now. Here, once again, is an antibody. There is a variable region which is different for every potential pathogen. Different variable regions on different antibodies. Antibodies bind to antigens with a kind of lock and key mechanism, similar to the active site for an enzyme and its substrate. The binding site is an area of about 110 amino acids, building blocks of protein, on both the heavy and light chains. There's a different shape on each antibody, which means it can bind to a different shaped antigen, a different toxin, a different bacteria. This gives the antibody its specificity. The rest of the antibody molecule is always the same, known as a constant region. Constant region. When bound to an antigen, it's known as an antibody-antigen complex. There are hinge regions which provide the antibody with flexibility, allow it to bind to separate antigens, one on each antigen binding site. So one Y-shaped antibody can grab hold of two antigens and kind of cross-link them, maybe sticking two bacteria together, for example, or coating the surface of a bacteria, clogging it up. So how exactly do antibodies work? Well, the antibody of the antibody is the antigen complex. It acts as an opsonin complex. It's easily engulfed and digested by phagocytes. So basically, these antibodies, when they're bound, they attract phagocytes to eat up the infection. Most pathogens can no longer invade the cell once they become part of an antibody-antigen complex, meaning once they're clogged up with antibody. Antibodies act as agglutinins. They cause antibody-antigen complexes to clump together, clumping up a bunch of bacteria, for example. Lymphocytes and immune response. A specific immune system is based on white blood cells called lymphocytes, the B lymphocytes that mature in the bone marrow, and the T lymphocytes that mature in the thymus gland. Main type of T lymphocytes are those helper T cells I mentioned earlier. And now let's go into a little bit more depth. They have a receptor on the surface known as a CD4 receptor. These CD4 receptors are on their membranes, and they bind to surface antigens on antigen-presenting cells. So they bind to these antigens on antigen-presenting cells. These are the antigens, well, we'll get to that. They produce interleukins, a type of cytokine, cell signaling molecule. Interleukins made by the helper T cells stimulate activity of the B cells. The B cells, this increases antibody production. B cells make and release antibodies. Lots of antibody. So this increases antibody production, stimulates production of other types of T cells, and attracts and stimulates macrophages to ingest pathogens. T killer cells, they destroy a pathogen by any pathogen carrying a specific antigen. They produce something called perforin that kills a pathogen by making holes in the cell membrane and making it permeable. Basically, if you plug a load of holes into a bacterial membrane, then water's going to flood in, the bacteria is going to swell, and it's going to pop. There are T memory cells. They live for a long time, maybe many decades, and they hold the memory of that pathogen. So once a pathogen is encountered, you make these memory cells, and they sit around in the tissues waiting for a reinfection, so they can mount a very powerful, very quick response. They meet an antigen a second time, they divide rapidly, producing a huge number of clones of killer T cells. T regulator cells, these are very important. The T regulator cells suppress the immune system, acting to control and regulate it. You don't want an overactive immune system. You certainly don't want an immune system that attacks your own cells. Your cells have got antigens on them as well. And you don't want immune cells that recognize your antigens as foreign. That happens, you get an autoimmune disease. So T regulator cells suppress the immune system by regulating it. They stop immune response once a pathogen has been eliminated. They calm it down. They make sure the body recognizes self-antigens, not to set up an autoimmune response. Basically, when your immune cells mature in your thymus, and you get all these brand new immune cells, all of them containing different antibodies, different receptors, they're all sort of tested on your cells to see if they'll react with your cells. And any that do, they're gotten rid of. They are marked for destruction. That's part of the regulation process. Interleukins, special molecule, are important in this control. So main types of B lymphocytes for plasma cells, they produce the antibodies. So when a B cell is activated, it will swell up, become a plasma cell, pump out antibodies. They can then circulate in the circulation, these antibodies will just then find any bacteria or infectious agent that has that particular antigen and go through the process we've already discussed. There are B effector cells. They divide to form the plasma cell clones. So B memory cells, they live for a long time, providing immunological memory. They're programmed to remember a specific pathogen. They trigger a rapid response when the pathogen carrying that antigen is encountered again. So cell-mediated immunity, cell-mediated response is important against viruses and cancers. In non-specific defense, the macrophages engulf and digest pathogens in phagocytosis, so in the neutrophils. They process the antigens from the surface of the pathogen, forming antigen-presenting cells, and then present that antigen to activate other parts of the immune system. So let's go into cell-mediated immunity. What happens next? Receptors on some helper cells fit the antigens. T helper cells become activated and produce interleukins. They're stimulated to produce more T cells rapidly by mitosis, which just means cell division. They form clones of identical T helper cells. They carry the right antigen to bind to a particular pathogen. The clone T cells may develop into T memory cells, give a rapid response if the pathogen invades the body again. They might produce interleukins that stimulate phagocytosis, cellular eating, produce interleukins that stimulate B cells to divide and to make antibodies and release them into the blood, stimulate the development of a clone of T killer cells that are specific for the presented antigen, and then destroy the infected cells. And for anyone watching this on YouTube, there's a little image here showing the process. A process of basically a particular immune cell, phagocyte, eating a pathogen, swallowing it, presenting its antigens on the surface so that helper T cells can then become activated and trigger the immune response. So humoral immunity. Here the body responds to antigens found outside the cell, e.g. bacteria and fungi, and to antigen-presenting cells. The humoral immune system produces antibodies that are soluble in the blood and tissue fluids. This is what the B cells are doing. Well, what one form of B cells are doing, the plasma cells. They are part of what is known as the humoral immune system. These plasma cells will release antibodies into the bloodstream, pump them out, all specific to a particular antigen. B lymphocytes have antibodies on their cell surface membrane. These are known as immunoglobulins, immunoglobulin M, for example. There are millions of different types of B lymphocytes, each with different antibodies. When pathogen enters a body, it carries specific antigens or produces toxin that act as antigens. So on the surface of some bacteria, there will be these antigens. Or if the bacteria releases toxins, they'll act as an antigen. A B cell with complementary antibodies will bind to these antigens on a pathogen or to free antigens. The B cell engulfs and processes these antigens and becomes an antigen-presenting cell. And this will then activate the immune system. So activated T helper cells bind to B cell antigen presenting cells. This is known as clonal selection. So a particular clone is now selected. The B cell with the right structure, the right shaped antigen. Interleukins are produced by activated T helper cells. They activate B cells. Activated B cell divides by mitosis to give clones of plasma cells and B memory cells. This is known as clonal expansion. The cloned plasma cells produce antibodies that fit antigens on the surface of the pathogen. They bind to the antigen and disable them, or they act as opsonins or agglutinins, which I discussed earlier. This is a primary response, can take days or several weeks to become fully effective against a particular pathogen. So it can take weeks for humoral immunity to become fully active, which is why vaccines don't work straight away and why it takes a while to get over a particularly bad infection. So humoral immunity, some cloned B cells develop into memory cells. If the body is infected again, B cells divide rapidly again to form plasma cell clones, produce the right antibody, and wipe out the pathogen very quickly, known as the secondary immune response. So a primary immune response is very slow because you've got to find the right B cell with the right antibody, and then it's going to make clones of itself, make plasma cells, pump out the antibody to get rid of the infection. But because you're left with memory cells that produce the right antibody, if years later you're infected with the same infection, then these memory cells will rapidly become activated. You'll make lots of plasma cells and pump out the antibody very rapidly, so you don't suffer the same symptoms as you did the first time. But of course, this can go wrong, which is where we get autoimmune diseases. The immune system can stop recognizing self cells and starts to attack healthy body tissues. This is known as autoimmune disease. It's not fully understood why this happens, but there is some genetic tendency in families. Immune system may act abnormally to a mild pathogen or body organisms. In some cases, T regulator cells just don't function properly, so they can't regulate the immune system. Around 80 different autoimmune diseases can cause chronic inflammation or complete destruction of healthy tissue. Lupus, for example. Immunosuppressant drugs may be used as treatments, but deprive the body of natural defenses, which means you're more likely to get infected if you take immunosuppressant drugs. So the immune system is very powerful, but it can go wrong sometimes. It can overreact, or it can react to the wrong types of tissue, your tissue, or it can underreact. It's going to be regulated very tightly. Okay, so that's all for now. Hope you enjoyed that particular podcast. If you want to know any more about me, you can follow me on Twitter. My Twitter handle is @markofd, that's @MARKOFTHE or you can go to my website, which is scienceontheedge.com. And if you want to donate to keep me motivated, then you can do so through my website by clicking on the donate button. Thank you for now, and I hope to speak with you all again very soon. Goodbye.